I'm Komal Yadav and today we are discuss the DNA. DNA stands for the deoxyribonucleic acid and we discuss the history of DNA. In 1869, Mr. discovered the DNA in the nucleus. Cells from the pus and the protein and acid molecule. It came into known as the nucleic acid. In 1926, Lewis proposed the tetranucleotide theory. It states that the nucleic acid is consists of the 4 nitridases. It gives the 4 nucleotides on hydrolysis. Rosalind Franklin used the X-ray crystallography to visualize the DNA structure. James D. Watson and the Francis Crick, co-originators of the double helical structure of the DNA. DNA is found in the living cells. DNA contains all the genetic information. Individual organisms have the unique DNA genome like the fingerprint. It contains the phosphorus in the form of the phosphatic deoxyribonucleic acid. It is the molecule that encodes the genetic information. Most DNA molecules are the double helical structure. Now we discuss the structure of DNA. DNA it is the right-handed twisted structure which contains the 10 nucleotides. Each spiral tank consists of the sugar and the phosphate group. It contains the base that is adenine, thymine, guanine and the cytosine. DNA contains the 3 main groups, that is sugar, phosphate group and the base. Bases is divided into 2 groups, that is pyrimidine and pyrimidine. Pyrimidine ring is consists of the 6 molecules, 6 member ring, thymine and the cytosine. Purine is 5 member ring. It contains the adenine and guanine. In 1993, Watson and Crick proposed the double helical structure of the DNA molecule. It contains of the 2 helical polynucleotide chain. 2 polynucleotide chain around the coil around the same axis from the right-handed double helix. This helix 2 chain are the antiparallel. Backbone of the chain is consist of the alternate sugar phosphate residue. Purine and the pyrimidine bases of the each strand are complementary to each other. Base pair is 0.34 nanometer and the complete helix of the DNA is a 3.4 nanometer. It contains 10 bases and the external helical diameter is 2 nanometer. It contains the 2 groups. The narrow grooves are called as the small groove and the minor groove, and the broad grooves are called as the major grooves. Pair between the purine and pyrimidine, that is the AT and the GC. There are 2 hydrogen bonds between the AT and 3 hydrogen bonds between the GC. Now we discuss the types of DNA. Nuclear DNA and the mitochondrial DNA. Nuclear DNA is located in the nucleus of the eukaryotic cell. It has the 2 copies per cell. Structure of the DNA, nuclear DNA is a linear. Then we discuss the mitochondrial DNA. Mitochondrial DNA is located in the mitochondria. It contains 1000 to 1000 species per cell. Function of the DNA: storing the genetic information, detect the protein synthesis, determine the genetic coding, and the directly responsible for the metabolic activities, evolution, hereditary, and the differentiation. It is the stable molecule which holds the information for the longer period of time. Now we discuss the how DNA technology used in the medicine. DNA testing can be used for the identify of the pathogens, identify the biological regimens in the archaeological diagnosis, stress disease, study the human migration pattern. Also, DNA is used in the diagnostic, new vaccine development and the cancer therapy. Advanced genetic engineering are mostly used in the medicinal preparation. DNA analysis can be used for the diagnosis of the hereditary disease, in investigation of the malignant process, in forensic medicine, for detection of the infectious pathogen. Two important methods approach the DNA analysis, that is the southern method in the DNA technology. Contain the enzymatic amplification of the DNA by the PCR. Cystic fibrosis is an example for the diagnosis. It is the serious hereditary disease. Its diagnosis improves after the introduction of the DNA analysis.